Hey man, what you working on?
Oh, nothing much. Just waiting for all my PCR to finish.
Oh, did you know we actually don't have to do PCR anymore with nanopore technology?
Son of a Nanopore technology? What the heck is that?
Yeah, so nanopore sequencing, it's a new approach in the sequencing of polynucleotides in the form of DNA and RNA that is done without using any PCR amplification. And
No way. Tell
Yeah.
me more.
Yeah, well, it works like that, and all you need is your handy dandy portable mini ion DNA and RNA sequencing device and a computer.
Hey, that's mine.
I need it for this. So to use the nanopore sequencer, you would just have to follow these 3 steps. Step 1, you place the flow cells into the mini ion nanopore sequencer. Just like, oh my gosh, that. Step 2, you load the samples onto the flow cell. And then step 3, you kick your legs back and relax as the nanopore sequencer does the work for you. But let's delve into how the nanopore sequencer does its science.
The nanopores are within the membrane wells of the flow cell. A tether is then used to guide the DNA or RNA sample into the nanopore, which then the nanopore uses current signals with different blocking degrees and blocking times to generate the sequence. This means that each nucleic acid model can be identified with their very own special current signal. The sequence is then formed and fed into the computer program's database.
So while we're waiting for our sample to be analyzed, why don't we explain to our fellow scientists why our product is the best of the best?
That is a great idea, Caleb. So first, nanopore sequencing is limited only by the length of DNA or RNA fragments present to the pore, and therefore can sequence entire repetitive regions, resolve structure variants, as well as differentiate between different isoforms. So size does not matter. The ability to sequence native DNA and RNA without the requirements for amplification also eliminates PCR bias and allows for the identification of base modifications, such as methylation alongside nucleotide sequencing.
Wow, that's pretty amazing. Another benefit is that the nanopore sequencer develops the sequence strand in real time, making nanopore sequencing unique, and that it is the only sequencing technology that enables direct real-time analysis of short to ultra-long fragments of DNA and RNA in full scalable formats. Advantages of real-time sequencing include the rapid access to time-critical information such as pathogen identification, the generation of early sample insights, and more control over the sequencing experiment.
Wow,
Yeah.
so much better. And the best part is, folks, let me tell you, it is very cost effective for every single lab at the low price of $1,000.
Oh wow, looks like our sequence has been done for a while now. Guess that's the benefits of real-time sequencing. Sequencing is important in the world of forensic science due to the importance that DNA plays in crime scene investigation. Because of the possibility of different genetic material being found at a crime scene, a DNA or RNA sequence can be used to help match or eliminate suspects from a crime. DNA and RNA is the present and future of forensic science. They're the future due to DNA being the genetic code of life. It is the instructions for building and operating an organism. RNA is primarily a messenger molecule carrying instructions from the DNA code to control the synthesis proteins, the building blocks of organisms. Sequencing can answer a range of biological questions, provide information on pathogen identity, genetic disease risk, or if a person was at the scene of a crime. Pretty amazing, huh?
Overall, nanopore sequencing technology is on the forefront of DNA sequencing. According to an article in the Forensic Science International Genetics Journal, it has an overall accuracy of 99.83% when analyzing 1,500 or less nucleotides, which is 15 microhaplotypes, which further justifies why this technology is amazing.
As a quick, accurate, and affordable option for DNA sequencing, there's no reason this technology shouldn't be present in every lab across the nation.
Call 402-472-7932. That's 402-472-7932. Limited time offer. Only available from nanoporetech.com. Product is made in the United Kingdom. Beware of imitators. May cause coolness, stress relief, and in some extreme cases, death. Use with caution. Call 402-472-7932. 402-472-7932. That's 402-472-7932.
The membrane wall.
Specular current signal. This sequence is formed and then it is fed into the computer program's database. This shit just doesn't work!